I am Dr. T.S. Chandrasekhar, founder, chairman of Medindia Hospitals and Academy. I am the ACG Governor, American College of Gastroenterology Governor, India region. I bring warm greetings from World Gastroenterology Organization as a member scientific committee, and I represent Tamil Nadu Dr. MGR Medical University as his honorary distinguished professor. The first two webinar series, I've seen 9,000 delegates from all over the world. That really inspired us to start the third webinar series on gastroenterology with the tagline, name the topic, the world experts are here. And we created a new Endo-Geo webinar series that really caught on very well. And we have scheduled this time series 3, 12 webinars from April 24 to March 25. The first webinar lecture was inaugurated by the President of American College of Gastroenterology, Professor Dr. Jonathan Lighton. And the next webinar is going to be on Barrett's esophagus and GERD. Nicholas Sahin and Sobana will be talking there. And I must thank my colleagues, Dr. Gokul Satya, Raja, and Prasad and TNS for their efforts to coordinating this webinar. And this webinar would not have been successful without the ambassadors we have all over the country, each ambassador representing one state in India, and we have international ambassadors from Malaysia, USA, Nepal, and various other countries. We have that, and I must be very thankful to them for propagating the advances in science to the next generation and the postgraduates and the medical students. And I must thank Dr. Beju for his contribution. And Tamil Nadu Dr. MGR Medical University has given credit hours for this all our webinars. And this would not have been possible without the academic partner, Micro Labs and Micro Knowledge Academy. I welcome our panelists. They are the eminent medical personalities in India in the field of gastroenterology. Dr. Sareen, Dr. Nageswar Reddy, Dr. Kru, Dr. Philip Agustin, Dr. Randir Sood, Dr. Amit, Dr. Mahesh, Dr. Usha Datta, and Dr. Pramod Garg, and Dr. Govind Makharia. And now today is the day that we are going to have the most eminent medical personality, none other than the Nobel laureate, Professor Dr. Barry Marshall, who is the discoverer of Helicobacter pylori, which has revolutionized the management of peptic ulcer stomach cancer. 50% of the world population is infected by Helicobacter pylori bacterium, and 50% of the Indians are also infected by the same bacteria. And unfortunately, most of them are asymptomatic, and there is a link between this bacteria and the stomach cancer. That's a great discovery. He changed totally the pathway, what we thought. In 1982, he discovered Helicobacter pylori, and he was awarded Nobel Prize along with his mentor, Robin Warren, in 2005. His discovery changed the medical perception for gastritis and peptic ulcer from physiological basis to infectious causes. No one would have ever thought of this one, and is the second most popular high-profile Nobel laureate in history. And I have a short video presentation about his work and his early period. Please enjoy this video presentation.
of the patients with ulcers had the bacteria. It's a given. These bacteria are probably the cause. Is not always ignorance, it's the illusion of knowledge. People already thought they knew the answer, but they didn't realise there was a lot of problems with that. In fact, they didn't have a cure.
There's something really good. We could even get a Nobel Prize out of this. I thought that'd be so bloody stupid.
When I recognised that there was a lot of resistance to the idea that these bacteria were harmful, I said, well, how do you go about proving it? So I said, let's infect a human. So I said to Dr. Warren, I think you should do it, Robin. Drink the bacteria. He said, oh, no, I'm too old, so you should do it yourself. And so I decided that I was going to have to take it. And on that fateful day, it was a bit scary, really. And people say, what did it feel like when you had to drink the bacteria? And I'd say, well, it's probably like taking your first bungee jump. You don't really know what's going to happen at the end. Theoretically, it's going to be safe, but you still have to gather up a bit of nerve to do it.
Who dares
I was quite
wins.
surprised after a week when I started vomiting, but quite delighted when on the 10th day I had an endoscopy and it showed that the bacteria had infected my stomach and I had developed gastritis. It was proven then that a healthy person could take the bacteria and be infected and get gastritis, and that was the underlying process, we thought, which was the lead up to developing ulcers. Probably around the world, hundreds of thousands of people had their lives saved by having Helicobacter treatment, and that's not mentioning stomach cancer, which also kills 1 million people a year, even today.
It all changed when the National Institutes of Health, NIH, held a 2-day summit in Washington, D.C. over the matter. They could no longer ignore the evidence. At the end of the summit, they released a statement stating that the key to treatment of duodenal and gastric ulcer was detection and eradication of Helicobacter pylori.
And I suppose it was only a matter of time before somebody in the Nobel Prize Committee had an ulcer, took the antibiotics, and they said, this is great, we must give a prize for this.
Thank you, Professor Marshall, for this wonderful journey. We want to know more about it. And when he has got Nobel Prize, what more award anybody can give? The only thing we can give is People's Award. The people all around the world who are watching this program, the medical doctors, pathologists, microbiologists, thousands of them are watching this program, along with that Academic Council of Med India Hospital and Academy, and my great panelists who is who in gastroenterology in India. We jointly present, and I take the credit in presenting these awards, as he was persistent in discovering Helicobacter pylori. I was also persistent with him in getting him to this webinar. I am very thankful to you, Professor Marshall, for accepting this honor. And we are honored to have you here, and please accept this award. Along with me, my colleagues are here, and my great panelists, my friends, Dr. Nage, Dr. Sareen, Dr. Philippe, Usha, and delegates from all over the world. We are presenting this. And this
it's going to look very nice on my shelf behind me in future webinars, I think. It's nice to see some people that I know there too. Hello, Dr. Reddy. I know we've had some discussions. Hope to meet you in person again soon.
And this medal to our professor. Virtually, we are garlanding you, and it will be courier to you. As I said, that will be courier to you. And I like to have a photo of holding this medal and the thing. Thank you, Professor.
Yes, that'd be great.
Thank you.
Okay.
And now the lecture session starts with this brief introduction. And I will be introducing the panelists later during the panel discussion. And the platform is for you. And over to you, Professor Marshall.
Thank you. Thanks very much for inviting me. And it's so nice to be giving a comfortable lecture in the evening, not too late. I know in the United States, often they want me to talk about midnight or 3 a.m. But Australia and India not too far apart time zone wise. It's very convenient, actually. So let me share my screen, which is H.Pylori, the journey of discovery, and further show another screen that I've got, just some homage to Dr. Warren. So this is Perth to Chennai via Singapore and KL. It's 11 hours and only 2.5 hours difference on the time zone because we're on the west. So if you know Australia, you're probably thinking about Sydney. That's another 3 hours. And this map, I think, has all the different countries proportionally stretched, so the area is the same. So I guess India is about half as big as Australia, but actually quite tall north to south. I'm coming from the University of Western Australia, and there we have the Marshall Centre, which is a microbiology lab. And, the University of Western Australia has a world ranking on one of the scores of 90. So in the top 100 universities. So maybe we'll get some Marshall Centres in India too, and that would attract me to come and visit a few more times and meet everybody in person. I know, I remember the times I've been in India, there's such great, smart students and very enthusiastic about science. So, it's very easy for me to do that. So we're going to talk about Alfred Nobel had Helicobacter, but before that, I just want to just pay attention to my colleague, Dr. Warren, and if I get him here in a window, there we go. That one, share. So I'm sharing a different window, and what I just wanted to say that poor Dr. Warren, he passed away last month. And so I wrote an obituary for him, which you can see online now. It's from the Medical Journal of Australia. It's a publication online that they have called Insight, and there's a lot of discussion, etc. So it just says that very few in 1979, on 11th of June, that was his birthday. It was his 42nd birthday. So he's about 12 years older than me. And he was examining a stomach biopsy specimen. He was seeing some gastritis because there's polymorphs or pus cells, if you like, in the tissue. And very few doctors in the world were interested in gastritis because it was common and believed to not cause illness. And I know this has been the predominant view in many countries, including India, and except in some special subgroups. And so if he looked, he was interested in bacteria, and he stained it with a silver stain, and he saw about 30 organisms on every single epithelial cell. So that really got him interested in it. He was doing some pen and ink drawings and planning a publication when I went on. So he was going to describe the pathology of it. And he looked at a number of other biopsies, and he found that it was quite common. And so that's when I met up with him in 1981 to find out if there was a clinical association with the bacteria or whether it was just inflammation in the stomach. And in those days, we weren't actually focusing on ulcers because we saw these bacteria in plenty of patients with no ulcers, just with the histology issues. And so there's a picture of Dr. Warren, probably taken about 2005, I suppose, after the Nobel Prize. And so I made in 1981, I made an appointment with Robin, and then for the rest of the day, I was educated about these curved bacteria, as well as microscope slides. He had detailed drawings of their various features. He showed me a few old pathology books in which gastritis was described, and he found a couple of rare cases where the bacteria had been shown in paper publications from other people, but then they moved on. They didn't really study it very well. So I collected the medical records of the cases and spent many weeks trying to see what diseases people had. And initially, I couldn't find any association with the bacteria. It was just the histology. And here's, of course, Robin Warren's first image of the bacteria. And that picture was published in The Lancet, but in black and white. But plenty of time now we've seen that. And there we are in his lab. This is 1984, after we had our first publication in the Lancet. And I love to tell this story about nerds who love technology. And Dr. Warren and I were both technology nerds. And so in those days, when you did a p-value, he was doing the p-value of the association, the chi-squared, between the bacteria and the inflammation, because we were studying gastritis in those days. And the statistical package we had was SPSS, and it would only measure p-value to 5 zeros, 5 decimal places. And we always used to get p equals 0.00000. But we really wanted to find out what the p-value was. And so Robin went out and he bought a Hewlett-Packard calculator, which could measure very large numbers. And then we did it exactly on a Fisher's test, I think, and we got a p-value of 10 to the minus 12. And I just wonder, we might have the world's record for a p-value in a pathology journal or in the clinical journal. So p equals 0.1201. We're very satisfied about that. Now, after that, actually, I started collecting Hewlett-Packard calculators as a hobby, and HP is the name of the calculators, and of course HP then became the name of our bacteria, so a lucky break for that. So I thought I'd just tell you a little bit about Robin Warren, and we had a great association for more than 40 years. And this photograph was taken on my phone, which is a Palm Trio smartphone, before iPhones, 2005, and that is actually Dr. Warren taking the call from the Swedish Academy to say that he'd won the Nobel Prize. And they said, we can't find Dr. Marshall. Where is he? He's not home. And Dr. Warren said, well, he's here having a beer with me. We always have a beer on Nobel Prize announcement day just in case. And so we were so happy finally we did get the Nobel Prize. So that's a rare photograph of Dr. Warren, 2005 in October. So let me share something different now. I'll go back to my lecture, and it's always fun talking about Dr. Warren, and he was a pretty great guy. So we go back to window, and I'm going to be sharing on my lecture now. So sorry about that interlude, but I think we've got a good amount of time here. So Alfred Nobel had Helicobacter. Most people in the world used to have Helicobacter in those days, and if we go through his biography, you can see here, Alfred Nobel suffered from poor health. He always had indigestion, headaches, and occasional spells of depression. So this is kind of a non-ulcer dyspepsia syndrome associated with gastritis, and present not in everybody, but about 25% of people with Helicobacter, I think, have these things. And now and again, you see a patient who has headaches, or it's sort of a vague depressive illness, like they don't feel 100% fit, and they improve a lot when you get rid of the Helicobacter. And so he, down the bottom, says that he had intense pain, angina pectoris. Now, people who had dyspepsia in those days used to be put on an ulcer diet, which is lots of cream and very bland food and no spicy food and not much meat or anything. So those people, they probably had very, very high blood lipids and all got atherosclerosis, and a lot of them died from cardiovascular disease as well. So probably indirectly caused by the fact that they had dyspepsia. And people always said that he was overworked or under mental stress, so that if they never actually could diagnose an ulcer on Alfred Nobel, but if they had done it, they would have blamed it on stress. They say he's a big billionaire, a tycoon, and obviously got a lot of mental stress about his business and everything. So you can see how stress fits into that ulcer sort of personality idea. So here's his silver stain, and the point that I would tell everybody, if you're not used to looking at gastric mucosa, usually it's complete. Normal mucosa is completely clean. There's no bacteria on it. They are above the mucus layer. And this mucus layer, remember, it's 100 microns thick, so it's 10 cell depths. So if this is the, lining of the stomach here, the mucus is the next, the 2 slides upwards. and it's usually clean, no bacteria in it. So bacteria cannot live in the stomach. It's too much acid. So we were very curious about that. What are these bacteria? How do they survive in the stomach? And someone said, oh, they're contamination, they're Pseudomonas, because Pseudomonas can grow in endoscopes. And that's why nowadays we, have a special scope cleaning machine and special staff to keep the scopes clean, not just Pseudomonas. But in the early days, everyone says, oh, it's just a contamination, just Pseudomonas. And if it came from the mouth, you see, it would have been many different kinds of bacteria, gram-positive, gram-negative, rods, hyphae, spirochetes, you name it. But this is a very consistent type of bacteria. You can see the beautiful Helicobacter shapes here. And in those days, we didn't know about Helicobacter. We used to call them Campylobacter, Campylobacter-like organisms. And of course, in those days, we'd only just discovered Campylobacter jejuni as a common cause of diarrhea. And so it's very, very common. And you would see like these kinds of bacteria in the feces of people who had Campylobacter enteritis. So it was a Campylobacter-like organism, but unusual to find something that could live in the stomach and be so common. So we know that now, but of course it was a big puzzle back there in 1981 and 82. So there we are in 1984. Now, 1984 was actually a key year. So that's one of the reasons we're having a conference on Helicobacter this year in Perth on 7th of October. But so it's 40 years since 1984, which was our first Lancet paper. And that's had maybe 4,500 citations, which is very nice. It's also the year that I did the self-experiment because I was very frustrated about people who didn't believe that Helicobacter was a pathogen. We had to solve this problem and move on with the research. And that was published in the Medical Journal of Australia in 1985. And that study also, we were looking and culturing Helicobacter. We did eradication pilot studies looking for some kind of treatment, and one of the best ones we found then was bismuth and metronidazole, and you know that that's still an important treatment. We invented the rapid urease biopsy test to give a quick diagnosis for gastroenterologists who had trouble finding someone who could culture the bacteria in those days. We did some serology, IgG, and we did the urea breath test, the first initial practice with the urea breath test. It wasn't on me. It was somebody in the head of the nuclear medicine department, as I recall, was the first one to do the urea breath test. Anyway, so that's a bit of nostalgia. You see, in those days, I was a penniless young registrar in medicine, and I used to have to buy my clothes in Kmart or somewhere, and of course they didn't fit very well. The trouser length was always a problem for me, but nowadays, of course, I got tailor-made trousers since the Nobel Prize gave me some spare money, and I don't have that leg showing all the time. It's a bit of a joke there. Okay, so let's say why is there so much resistance to new ideas in medicine and science? And this is something that a lot of people don't realize. So let's see what's that all about. Well, the person who knows about this or knew about it was this guy, Daniel Boorstin. He was the librarian for the US Congress, a historian. He had a lot of publications of books. And he said the greatest obstacle to knowledge, it's not ignorance. It is the illusion of knowledge. So everyone's there. We already know the cause of ulcers. Please don't make us learn more causes. So that's the situation. And people couldn't believe that they had overlooked something that was important like H. pylori. And I show this picture because these days in Australia, we've pretty much wiped out Helicobacter, except immigrants and Indigenous people, I think. But, in most of the cities like Melbourne and Perth, we hardly see duodenal ulcers and peptic ulcers anymore. And so I have to show the medical students this one on the left is a duodenal ulcer, and the one on the right is a gastric ulcer. You can see how that could go right through the stomach wall there and be quite deep and perforate through. So these things are becoming rather rare. And nowadays, so many ulcers in Western countries are caused by the non-steroidal anti-inflammatory drugs rather than H. pylori. But H. pylori and non-steroidal inflammatory drugs, they're the two big key causes of ulcers. So here's a statement from an article about Helicobacter, but it says established gastroenterologists found it hard to accept the teachings of the two Australians. Not only were we completely unknown, but we also seemed to be supremely confident that their hypothesis was correct. And we did an intensive literature search and studied, all the histology literature and microbiology going back 100 years. So we found that other people had seen these bacteria and made perhaps the wrong conclusion from them. But it wasn't like it was a new discovery. There was plenty of evidence that these bacteria were all over the world. And even some sort of related to peptic ulcers, you'll see in a minute. So we knew that bacteria had been seen before. And the person that I saw that was the first one was Giulio Bizzozero, who was in Turin. And I think he published in the German literature, so a lot of people didn't know about this. Of course, I didn't see the German literature 100 years ago. And he said, well, actually, Giulio Bizzozero was famous because he was the person who proved that all dividing cells must have a nucleus. And everyone said, not true, because, of course, red cells don't have a nucleus, and there's millions of them. But he said, no, they come from nucleated precursors in the bone marrow. So that's why he was famous, actually. And he was in the anatomy books. But he also used to look at the stomach of cats and dogs. He said, guess what I found? Acid-tolerant spirochetes are living in the dog stomach. So just be aware that you can catch Helicobacter, a dog type of Helicobacter or cat Helicobacter, from common animals like that who often have a Helicobacter infection, but it's not exactly, usually not the human one, but it can make you quite unwell. So he said, I saw spirochetes in the dog stomach, and he actually wrote a thesis about this. And so here's his illustration. There's a color print. I don't know, it probably cost the equivalent of thousands of dollars to put color in your thesis in those days, because you had to have it on a special page. Anyway, so here's a parietal cell. As you know, parietal cells there have a round nucleus and a big nucleolus, stained red. They're called oxyntic cells. And of course, here in the canaliculi, where the pH is only about 1.5 or 1, you see all these spiral bacteria here. And he said they're spirochetes. So this is what if you look very closely here, you see there's about 12 wavelengths there, and that is the Helicobacter of the dog. It might be called Helicobacter felis or Helicobacter helmanii if it's seen in humans. So he said they must be acid tolerant, or else they could turn off acid or secretion, or both. And now we know some mysterious things about Helicobacter. It can turn off acid secretion in the acute infection, and of course it's acid tolerant. So that was 100 years ago, but of course we didn't have the technology then, like culturing methods, microaerophilic. We didn't have endoscopy. So that was about as far as you could go in those days. So Dr. Warren and my colleagues said to me, Barry, we should do 100 patients and just see what this all means. Is there any disease association with these bacteria? Excuse me, I have my coffee. And so we did it. We did 100 patients, and you see I did the endoscopy correlations, and I didn't know the microbiology or the histology. It was prospective and it was blinded, but we just did a biopsy on everybody who had an endoscopy that month. And here you see we had 100, and in those days, in 1980, I'm trying to tell you what, it was 1984. No, I think it was 1982. Yeah, 1982, that's right. Sorry. So in those days, we didn't have computers. So we always used to put 100 patients in our study, because then when you got 58 patients with bacteria, as you see the lower square here, that's 58%. There's no calculation required. And so we didn't need computers if we had 100 patients. So anyway, so there you see, we're very surprised. 58% of the patients had the Helicobacter. And then of these patients, there's different diagnoses down here. And if they had a duodenal ulcer, that's 13 patients. All 13 had the bacteria, 100%. And that's a p-value 0.304. And gastric ulcer, as I told you, gastric ulcers we know can have a non-steroidal anti-inflammatory drug as a cause. And that's what we found. We found that out of 22, 18 had the bacteria, so mostly Helicobacter, and the other 4 patients were non-steroidal anti-inflammatory drugs mostly. So that's the kind of data we had, and we said, well, this is exciting. We've got to study this a bit more, and we tried to get some research grants. We ultimately did get a National Health and Medical Research grant, which was funded in about 3 years later. But anyway, we did a lot of research in the meantime and submitted our paper and everything. But then we asked this, we had a lot of trouble getting the paper accepted. Even the letters to the Lancet took 6 months. So the question was, would the world accept the new cause for ulcers on the basis of data from only 13 patients? You know, the ulcer drug treatment business in those days was really starting up with H2 blockers, was already a global business of $3 billion. And of course, we were a bit cocky, if you like. We're saying, oh, all those pharmaceutical companies like Glaxo are going to go broke because we would just take antibiotics in the future. So, of course, they were a bit unhappy about that. And so, obviously, of course, the answer is no way. The medical fraternity is not going to accept this idea unless they've tested it many different ways, different countries over a few years. And so there was a lot of hard work was necessary after that. And we had a lot of trouble getting papers accepted, as you know. So here's our first attempt at publication, 1983, and this is in a local Australian College of Gastroenterology meeting in Perth. So the Australian gastroenterologists there, and we submitted our paper, and they said, Dr. Marshall, we regret your research paper was not accepted for presentation. The number of abstracts we receive continues to increase, and for this meeting, 67 were submitted, and we could only accept 56. And so they said, your paper's in the bottom 20%. And they said, Dr. Marshall, you can't get an oral presentation. And by the way, we're not even going to give you a poster. I said, what? And they said, we're not giving you a poster. It's not good enough. It's too unusual. And so you see, we didn't get a poster. But that was very difficult. And any young researcher might have that experience. I suppose we all have a lot of rejections when we're first starting out. I even get a lot of rejections even now. But it encouraged us to try harder and present the paper internationally. And so there were a few more thoughtful international researchers doing ulcer research. And we contacted people in the United States and in UK and in Holland. And we've ended up with a sort of a guerrilla warfare group, if you like, of microbiology-related gastroenterologists and scientists who were very interested in the bacteria, and things took off from there. So there were many causes of ulcers which were already known, but these were false. That's the illusion of knowledge. Stress, that was the big one. False. Bad diet. False. Alcohol, that's false. And genetic, well, you know, a little bit of genetic. People make more acid if they are, you know, related to people with a lot of acid, I suppose. But in actual fact, genetic means in those days, we could see that it ran in the families and you inherited some kind of thing from your parents. But of course, you see, if you lived in a village with contaminated water supply over hundreds of years, you could be getting some kind of cancer or some kind of illness, which apparently was running in families. And it was not genetic, it's environmental, but it's been there for several generations. And so that's what Helicobacter was. It's an environmental infection, and you catch it from your mother. And we'll talk about that a bit in the future. So Robert Koch had this problem with tuberculosis. Nobody believed that tuberculosis was a serious thing because so many people were coughing up these bacteria, acid-fast bacteria, and they didn't die from it. They had a bit of bronchitis. They didn't seem very bad. And so they couldn't believe that TB caused a fatal illness, you see. So Robert Koch, he did these experiments on guinea pigs. And he said to prove the bacteria are harmful, it should be infected into an experimental animal. Then if TB develops in that animal, it's proven that the bacteria are harmful. And of course, every medical student knows this is what's called Koch's postulates. And amazing coincidence. So we did my experiment. I'll tell you about that next. In 1984, and Koch's postulates were published in 1884. So it's exactly 100 years later I took the bacteria. So another amazing coincidence. Okay, so unable to find a suitable animal model, Marshall must experiment on himself. So for more than 6 months, Warren and I were infecting animals with these bacteria, but the infection wouldn't take hold. We couldn't, rats and mice and rabbits and guinea pigs and full-grown pigs, or half piglets actually, none of these animals were susceptible to Helicobacter. So we said, well, look, we need a human experiment. And after some discussion, we decided that I should be drinking these bacteria to see what happens. Would I get an ulcer? Would I develop gastritis? We didn't really know. And we thought probably nothing would happen, but the histology would change. Maybe over a few years, I might develop a tendency towards ulcers. So we were quite surprised what happened, actually. So here's a comic produced, would you believe it, from the Abbott Company in Chicago, who make clarithromycin. So clarithromycin was the best antibiotic for Helicobacter, and they were trying to get some publicity. So they made a comic, and this is it. There I'm drinking the bacteria. My lab tech says, Dr. Marshall, you're crazy. I'm saying, well, there's no other way. So I couldn't see any way forward until we had some more data on the pathogenicity of the Helicobacter. And so after I drank it, I felt a little bit of dyspepsia, a bit of fullness, not digesting my food properly for a few days. And then I started vomiting. Every morning I was vomiting. And there was no acid in the vomit. So it's very unusual to have a vomit and not be able to taste acid. So this was like just water was coming up. So a lot of watery secretions with no acid. And we know that's the acute achlorhydria or acute hypochlorhydria that often occurs, we now know, in Helicobacter. So I'm there, well, I'm not feeling very well. And after 10 days, Dr. Warren takes a biopsy, says, Dr. Marshall, your stomach lining is dangerously inflamed. So in actual fact, that's a bit of exaggeration. I just had some redness. I didn't have any ulceration or erosions or anything. And I was very uncomfortable during the endoscopy. So I know if you're a gastroenterologist, just be aware that people with gastritis and Helicobacter, they often are very, very sensitive. And when you start touching the antrum and pylorus area, they'll have uncontrollable gagging. It's very sensitive. So I always have endoscopy with no anesthesia because I've got a lot of work to do. I can't be sleeping around all day. So if you really do some research, it's good to have anesthesia these days, because then you can spend your time carefully looking at the stomach, taking some good biopsy samples. But in those days, we noticed a few things like patients who didn't tolerate it. So here's my silver stain of my stomach on day 10. You see the Helicobacter there growing there. And this was published in the Medical Journal of Australia, this photograph. And you'll see these, if you know the histology, there's no mucus cap on these cells. So the epithelial cells are quite abnormal, bit kind of primitive almost, and they've lost all the mucus layer. So you can see there's something that loses the mucus layer, interferes with the mucus. It's got phospholipase, digests the mucus layer. That is a good way of predisposing to peptic ulcer. But of course, the inflammation underneath and the polymorphs coming up through the mucosa are also toxic, releasing all kinds of things to digest the bacteria and damaging the mucosa. So that's the concept we have of peptic ulcer nowadays. So we proved that Helicobacter can infect a healthy person. It's the cause of gastritis. And we realized then that this was a disease affecting more than half of all humans. So it's been decreasing during the 20th century, but certainly it's 40 or 50% even now. And in many countries, India is probably 50% plus. Africa, South America, China is probably about 40% on average now. So it's going down, and part of that is raised standard of living and more understanding of hygiene, education. All those things are important. So the question again, how can a bacteria survive in the stomach and resist acid? So this was the beginning of our thoughtful part of our basic research then. And we realized then that H.Pylori has a urease enzyme. And it wasn't me who discovered this. It was, I think, Dr. Langenberg in Holland. And she noticed that it makes urease enzyme. So with urease, you can produce ammonia and protect yourself from the acid. And so there's a H. pylori inside the organism, pH 6, around the organism, pH 1. So that is a terrific mechanism for tolerating acid for long enough to get onto the mucosa. Now, here's the reaction of urease enzyme of H. pylori. I like to put some chemistry in there for the basic scientists. And you see urea. Urea is 2 ammonia molecules. Ammonia is a waste product. Your body is exporting it to the kidneys as urea. And also it's a CO2 molecule in the middle. So when you split urea with urease, it takes up a hydrogen ion. So each molecule of urea consumes a hydrogen ion. And then on the other side of the equation, you've got ammonia and bicarbonate. So you can see you've got an alkaline environment around the bacteria. It's a wonderful reaction, incredibly fast. And H. pylori makes more urease than any other organism. And from your microbiology, it might be Proteus mirabilis lives in the urine and makes urease, but probably only 10% or 1% of what Helicobacter does. So very few organisms make so much urease. That's the year I developed the urease test. I think it was about 2003, 2004, 2005, there. And here's this is the first actually commercial diagnostic test for Helicobacter. It's the urease test, and now millions of those tests are done all over the world. Gastroenterologist can take a biopsy and put it into the gel, and if ammonia is produced, it becomes alkaline and goes a red color, just like litmus. And you'll see that this in Australia on that particular day, which is probably about 10 years ago, we only had 20%, 1 out of 5 patients positive for Helicobacter. So interestingly, you get that result probably in about 15 minutes before the patient goes home. You could start treatment if you wanted to. Or you could culture the Helicobacter out of the gel, and culture rate's about 90% from these. And so your microbiologist would be very happy to receive a CLO test with a red gel. If you can get it to the lab in an hour or so, have someone take it down, they should be able to culture the H. pylori from that. So it's a good way to get your lab skills up. And they should be able to reach a 90% culture rate from the CLO test, if you like that. So then after that, we looked at urease in the stomach again, and we needed a non-invasive test. And so your family doctor, he didn't want to be sending all these patients to gastroenterologists to have biopsies. Family doctor likes to do everything himself. So you see, a family doctor likes to do the urea breath test. And now, same thing, millions of breath tests are done around the world. And this is the one that we have in Australia and the USA. It's called Pi test, if you like. But the lady's taking a capsule there, and she blows up a litre balloon. And in that balloon sample, you can see the isotope of carbon appearing in the breath. And the carbon-13 and the carbon-14 breath test could be done pretty similarly with this kind of technology. So I like to talk about the breath test because, excuse me, I had a couple of patents over the years back in the 80s, and they expired about the year 2003 or 2004. So I can talk about them now because I don't get any royalties from breath tests. Everybody's doing generic ones. But anyway, urea breath test, here's an overview. So the carbon-13 urea breath test, about half of them in the world are carbon-13. The thing about that, you have 2 samples. You have a baseline sample because everybody's baseline varies depending on whether you eat a lot of corn or different kinds of vegetables. So you need to measure the baseline sample before you start. And then 20 or 30 minutes later, you do the measurement sample. So that's the second sample that you have. And you want to see if it increased by a certain amount, by 1 or 2% concentration of the carbon-13. And so it's a very accurate test. It's done with infrared spectrometry, mass spectrometry, and so it's a really clever instrument which was actually used for measuring pollution from exhaust pipes in cars in Los Angeles. From General Motors, I think, invented that technology. I'm not absolutely sure, but anyway, so it's a laser gadget inside a little box, And it's a 30-minute test because of the fact you've got to have a sample and a follow-up sample. So that's right. And also it's 75 milligrams of urea. So, it's a capsule full of urea, carbon-13 urea. I'll just make the point here. It's 75,000 times more urea than in carbon-14 test. So you can see this would take relatively longer for the stomach to digest it, or the H. pylori to digest it. So if you look at carbon-14, it's a little bit of a contrast. It's just one sample because the baseline is always 0. So anything in the breath test that's carbon-14 must be coming from the Helicobacter. So baseline not required. And so that you're able to do it in a 10-minute test. And that's because it only requires 1 microgram of urea. So it's 75,000 times less urea. But, as soon as that capsule breaks open, that urease will react, I don't know, in about 1 minute. But if you actually do continuous breath sampling on people with a C14 urea breath test, 3 minutes, they're usually positive, 3 or 4 or 5 minutes. So we'd like to go 10 minutes so that we got everybody, but some of them react very, very quickly, as you know. So why is that? So think about it. In the stomach, you've got the lumen at the top here, pH is 2, and then you've got the mucus layer, and then to scale down there, you've got the epithelial cells and the mucosa. So the pH at the bottom of the mucus layer is 6. You see, urease is denatured in acid. So you can't actually have urease, active urease in that part of the stomach. So what we did in the Pi test was try to get the urea isotope down on the mucosa, where in the mucus layer. So the H. pylori loves to be down there stuck on the cells and not being killed by the acid. And so that in the capsule on our test, you see the caps are starch beads, and they've got C14 urea on the very surface there. And you'll see a reaction then with the H. pylori, and you're getting ammonia in the stomach, you're getting CO2 in the blood, and that's why it comes out. So all breath tests are the same. Urea breath test example of C14, but C13 is similar. So you could say you give the isotope, it goes into the stomach. If the H. pylori is present, you see the urease is going to break it down, and the isotope will then come out in the breath as the CO2. But if there's no H. pylori, the urea stays intact and goes into the bloodstream and is excreted in the urine. So if you do a urine collection, all the isotope comes out in the urine pretty much if you don't have H. pylori. So, if you want it to be a backwards person, not as accurate, and could wait a long time, you could do a 24-hour urine collection and diagnose H. pylori, because there would be urea present in the urine with the isotope. I'm very proud of that. That's one of my patents, May 16, 1989. So it takes 5 years to get a patent, unfortunately. And then it takes 20 years. So by 2005, it was gone, and lots of people were already doing it. Now, I like to talk about this a little bit because I want to remind you that the carbon-14 urea breath test is completely safe, and it's been vetted by the NRC in America and the FDA. So several studies in respected journals and millions of tests done since 1997, approved by the US FDA. The C14 urea breath test is 100% safe. And we know this from physics. So I now have a little personal rant. And we talk about, in 2024, we believe in physics. And I know, so many Nobel Prize winners have come in mathematics and sciences have come from India over the years. So we believe in physics. And I like to put these space pictures up there. So here's a picture of the moon and all the different things that have landed on the moon, some successful, some not. But I like the Chandrayaan one from India, 2023 August, that landed, and apparently there's this little robot driving around and taking photos of its spaceship and various things from the South Pole. So, you can do that because it's science, it's physics. You can make a calculation, you know exactly what's going to happen, and you can emulate it. So I would say we believe in physics, and so that's why we can do breath tests. So let's go and talk about a few details. It can be measured, it is real, and it's true everywhere. So let's go on here. So if you remember chemistry, Amadeo Avogadro, he was Italian, and he invented Avogadro's number. So carbon, P6N6, 6 protons, 6 neutrons, has an atomic rate of 12 grams per millimole per mole. And a 65 kilogram human has 18.5% carbon, which is 12 kilograms of natural carbon. 12 kilograms is 1,000 moles. And from Avogadro's number, that means every person has got 6 trillion squillion carbon atoms, whatever that is. And one carbon atom in 1 trillion is carbon-14. So you've already got some carbon-14 in your body, 1 by 10 to the minus 12. So all humans contain 6 times 10 to the 14 atoms of carbon-14. You have it in your body from the day you're born. That's 600 billion atoms. No, sorry, 600 trillion atoms of carbon-14 are in your body, no matter what. So if you say, okay, natural carbon has this activity, 13.56 dpms, and 12,000 grams in a human means 162,000 disintegrations per minute, if we could get all your carbon-14 and measure it. And per second, it's 2,700. So you have these radioactive counts in your body every second. And a standard unit is kilobecquerels, 2.7 kilobecquerels. So since this amount is already inside you, it must be harmless because you have that all your life. So this is the kind of thinking we've got. And the dose in the carbon-14 urea breath test is 37 kBq. But in one day, 90% of it is excreted. It leaves 3.7. So you see the amount that we're giving you is 3.7. And in your body already, you had 2.7. So this is very, very close to background. So how does that compare with other sources of radiation? So natural radiation, we're talking microsieverts there, which is the international standard, except in the US. But your own carbon-14, you get 50 each year, just from your carbon-14 in your body. Your potassium in your body, and we'll talk about potassium in more because it's pretty funny. You've got 400 each year coming from the gamma rays from your potassium in your natural body. Urea breath test, 3. And plane travel in 2 hours gives you 6. So every time you go any kind of plane flight, you're getting 3, 4, 5, 6 breath tests, 20 breath tests if you come from Australia. So here's something about the radiation limits by the NRC allowed and natural radiation limits in millirem. So this is millirem. The units are different, but it's the same kind of thing. This is in American data. So here's a nuclear worker in a nuclear reactor or somebody like that. He's allowed to get 5,000 each year. A whole body CT gives you 1,000. Average US annual dose of radiation, including everything, X-rays and also natural radiation, is 620. So that's probably the same in India. Average natural background in US is 310. Annual public limit. If you live next door to a nuclear reactor, how much are you allowed to get each year? And you're allowed to get 100. From your body, you get 40 cosmic rays. Everybody's got cosmic rays shining on 30. Chest X-ray is 10. Drinking water is 4. A transatlantic flight is 2.5 millirems. And the urea breath test is so small, it doesn't show up on any of these graphs. It's like 0.3. So it's like 8 breaths. So 3 there. So it's 8 breath tests is a transatlantic flight. Okay, so now that we've talked about this, here's the interesting bit. I asked ChatGPT about this because people have said, oh, yeah, bananas are radioactive. And, you know, you can put a Geiger counter on bananas and find increased counts, no problem. So bananas contain a small amount of radioactive potassium, and the radiation dose from eating one banana is known as the banana equivalent dose. So everybody talks about this a bit tongue-in-cheek, which is approximately 0.1 microsieverts. And so to get a dose of 3 microsieverts from bananas, you could eat 30 bananas. Now, you wouldn't really eat 30 bananas in a day, but you could easily, you know, effectively have a breath test each month by having a banana every day, which is probably millions of people. That's actually the world's most common fruit, is my understanding. So you would need to eat 30 bananas to receive a radiation dose of 3 microsieverts or 1 breath test. So that's why I'm not worried about giving breath test to anybody. I don't have to do a pregnancy test. I don't worry if as a child is like old enough to do a breath test in a good way, swallow a capsule. I can do it on children, anybody. And so I don't like to see warnings on the C14 urea breath test or the C13 breath test. Okay, so let's get back to basics now. How does H. pylori do its harmful damage? All H. pylori causes gastritis, and you see on the left, normal with the bacteria, and then this is gastritis. Now, this is actually quite a severe case, and it's actually from a gastric ulcer patient. They tend to have worse gastric inflammation, but you see all these little spots. They are lymphocytes, plasma cells, mononuclear cells. It's there. Your stomach is like trying to turn into a lymph node and fight the bacteria, but they can't get rid of the bacteria because they are not invasive. The bacteria are there on the surface inside these glands, and you would see, like this picture on the left here, if you put a silver stain on that. So that's the normal thing. And I know that in India, 50% of the population at least, but some places, if you're affluent in a clean water area, it might be 30%. If you're the average person in India, the average is about 50%. And if you're in the farming areas in India, it's probably more than that. It just depends on, how the hygiene is. And also, you catch H. pylori from your mother, and we'll talk about that again in a second. Gastritis can cause, some people have a high acid level. So the funny thing about ulcers, people who get ulcers are the healthiest people. The people who eat meat, they're big Americans, they make a high acid level, and they have high acid in men more than women. So that's peptic ulcer, about 10%. So 80% of people don't have very many symptoms, and they have a normal range acid level, and they may be dyspeptic, like they have a sore stomach from the gastritis. But often they just think they are normal. And but sadly, some people have a low acid level, and I think that's pretty common in Asia. And I'm conflicted about India. We can get into a discussion about this, but people who eat less protein and less meat, and so vegetarian diet might tend to keep you in the low acid level. And these people have no symptoms because the symptoms in your stomach are nearly always caused by acid in the wrong place or too much. And so these people, because they have low acid, they probably have a cancer risk. So how do you catch H. pylori? And I'm trying to speed up a little bit, and I'll just go back there. So in China, they did this study on about 8,000 couples, and they wanted to see who had H. pylori in the family. So they just breath tested everybody. And they saw that in 33% of families, there was no H. pylori in nobody. 24%, the husband was the only one infected. 20%, the wife was only infected. And in 23%, everybody in the whole family was infected, both. So then they looked at the children from these different families and say, well, where does H. pylori come from? Who do the kids catch it from? So firstly, they looked at these children with uninfected parents, and only 12% of them had H. pylori. So they didn't catch it from the parents. They got it from the environment, other children, I don't know, drinking water, whatever it could be. So that is the natural background rate of H. pylori, about 12%. What about if the father had H. pylori? Oops, excuse me. If the father had H. pylori, only 11% of the children had H. pylori. So the father doesn't seem to be spreading it to the children. Doesn't make any difference. They're still at background level. What about if the mother? Now, if your mother had H. pylori, you've got a 25% chance of catching H. pylori for each kid. And the children amplify it between each other, of course. So ultimately, with both parents infected, you get up to 52% in this study. So that's if both parents are infected. You catch it off your parents, probably your mother. But if no parents are infected, 12%, that's the background rate. Now, if it was spread by eating and, food utensils, chopsticks or knives and forks, things like that, then people would be catching it off their father. But apparently that doesn't happen. And I would say mothers are probably cleaner than fathers, but you don't catch it off your father. So that's evidence against, hygiene and your eating habits as the spread. And probably you catch it off your mother when you're a small child. She's kissing you, she's feeding you, she's taking the spoon and licking the bottom of the spoon, put it in the baby's mouth. So there's a lot of saliva connection with the mother, and sometimes the H. pyloris will be in the mother's mouth, and we can show that in some individuals. And in China, they told me that the mother always chews up the fish, takes the bones out, and then feeds the chewed up fish to the baby. So in China, that's how everybody got H. pylori. So there'd be things like that, pre-mastication of food. In many, many countries, it's something that was phased out in England about 100 years ago, but in most countries it continued on. So how does Helicobacter lead to stomach cancer? That's one of the issues. And so I just remind you, it's very interesting. This is a bit of a paradox. So this is Asia. So there's Korea, Mongolia, and Japan. We know that strains of Helicobacter in these Asian, Chinese countries are very toxic. They make the Kage toxin. Nearly 100% make the toxin, and of the worst kind of the toxin. So that causes more inflammation, and so we can see that we'd get more cancer. So, but actually, we go down here. It's very interesting that down here, Bangladesh, Australia, New Zealand, and India, about on the world average, 4.5%. Sorry, 4.5 persons per 100,000 per year. So it's not particularly common in India, but really gastroenterologists see plenty of it. Now, we then did a bit of a literature search, a Google search, if you like, and you'll see that in this Times of India report, I'm not sure where that data came from, but you see they said stomach cancer is one of the most common causes of death. So certainly when you get stomach cancer, it's usually diagnosed late and you'll die from it. So in this report, stomach cancer is a bit of a worry in India. And of course, it happens when you're older, above the age of 50 or 60. So India is still a pretty young population. And so as people live longer and get older, your mother is going to get stomach cancer maybe. When she's 70 or 80, she's just starting to retire and have a bit of relaxation, and after the economy is improving, and lo and behold, she gets cancer. So that would be a tragedy. And then we've got some other gastric cancer rates. We see India coming to 4.5. World is 9.2 average. And some interesting places down here. Brazil is much more. And Vietnam is a bit of a hot spot we've noticed. I was recently in Vietnam as well. So a lot of interesting work to be done on the epidemiology and molecular studies on Helicobacter in India and around the world. So I'd encourage everybody to have a think about this. Maybe if there was a conference on it in India, I would certainly try to go to it, because I think India might be the one of the keys to understanding what causes the cancer and whether there's dietary things infecting it. So I mentioned that bone marrow stem cells may cause gastric cancer. And this is a study published in 2004. And it's a beautiful study by Jean Marie Houghton and Dr. Wong in Boston. And firstly, so the concept is that cancer comes from bone marrow derived cells. So how do you prove that or test it? So they're female mice and male mice. Now, these transgenic male mice have got green fluorescent protein in all their cells. And of course, they got the Y chromosome as well, by the way. And so here's a female mouse. And so we irradiate that mouse. So the poor mouse loses her hair, very unfortunate, but no bone marrow. So that mouse needs a bone marrow transplant to stay alive. So we give it a transplant from this male GFP mouse. So now this female mouse has got male marrow, and we can infect that mouse with those dog and cat Helicobacters. And here you see Helicobacter felis in the mouse. And after about 6 months, these mice will develop some kind of stomach tumors. And we can study the stomach tumor, then the cancer in these mice. Where did it come from? So looking at the histology and using fish to see those fluorescent genes in the control, you see on the left side there, we've got just normal mucosa and no fish, no green spots here. When this mouse developed adenocarcinoma, we could take the same section of the adenocarcinoma and then study it And there you go. We see GFP inside the nucleus of these cancer cells. Okay, so green fluorescent protein came from the stem cells. And then look also for the Y chromosome. And lo and behold, this cancer has got Y chromosomes in it. So it's come from the stem cells from the bone marrow transplant. So the concept in cancers to some extent is chronic inflammation over your whole life. You get a repair process which attracts stem cells, and then you are very susceptible to carcinogens. So in this study, they used Mongolian gerbils, which do catch Helicobacter and develop gastric cancer. And they're comparing salt and carcinogens, because a lot of the dietary studies have looked at carcinogens and salt in the diet. And there's something there that means that it can be harmful. So look down here. Firstly, we look at gerbils getting salt, sodium chloride. In this column, look on the right side first. So this is the titer of antibodies, and this is correlating with the inflammation. And then on the left side, you see the number of those gerbils that got cancer. So with the salt by itself, nothing happened, no inflammation. So they might get ulcers or something, but it's not inflammatory gastritis. Helicobacter, they get a bit of inflammation and they'll have antibodies, but they don't develop cancer just from Helicobacter by itself. Helicobacter and salt, you'll see the antibodies are higher, which means the inflammation is worse. So salt aggravates the inflammation of gastritis. And then the carcinogen, MNU, no cancer, no inflammation. MNU plus salt, nothing. MNU plus H.Pylori, look, now we're getting the antibodies and we're getting some cancers. So carcinogen aggravates Helicobacter gastritis a bit more. And carcinogen plus Helicobacter plus salt, all three, you see a high titer of antibodies, and about 50% of those gerbils develop stomach cancer. So you see, the moral of the story is this: H. pylori makes everything else much worse. So a little bit of carcinogen, then we can worry about that. So Dr. Chandrasekhar, how are we going there for time? I know we started a little bit later. I've got about another 5 or 10 minutes, I think, if people can stay around.
We have around 10 questions. Panelists are waiting. So how do we go about it? You want to complete in 10 minutes? I request my learned panelists.
Why don't we just, I'll go very quickly and just skip over a few things. Okay, the importance of eradicating H. pylori. And this is a Korean study, which I encourage you to look at, family history of gastric cancer. So if you had someone in your family who had gastric cancer, your risk is 4 times or more of the H. pylori risk. And in a placebo-controlled study, the risk of not eradicating H. pylori, 5% people got cancer. These are relatives of cancer patients. 5% got cancer in 12 years. If you eradicated the H. pylori, it's about 2.5%. So you don't cure the cancer risk by curing H. pylori. You decrease it by 50%. And gastroenterologists should still be careful about looking at the mucosa and trying to figure out who's at high risk, who's still going to be getting stomach cancers. That's one thing. So the take-home message again: get rid of H. pylori as soon as possible before this cascade of malignancy has seeded itself. So in the future, everyone says, oh yeah, Dr. Marshall, how do you treat it? We can talk about that, but we want to encourage everybody to use the 21st century strategy: novel technology, precision diagnosis, specific therapy. We don't give the same antibiotics to every single person anymore. We give you personalized medicine. And this is going to result in less antibiotic use, shorter courses, and no more superbugs. And this is very important because people only take the antibiotics when they need them. So how do you solve that problem? If we look back actually on Helicobacter, I just want you to look at this red line here. That is the antibiotic resistance in China. And here you see metronidazole, 60% plus was resistant. Here we see clarithromycin, 60%, 50 to 60% resistant. And then this one, levofloxacin, well, the names have moved the wrong way, but levofloxacin, 60 to 70%. So those antibiotics, we would like to know if a person has resistant organisms before we treat them and not just see failures. So here's my simple strategy for treating Helicobacter. And just remember these rules. These antibiotics can be used again. So a lot of treatments, you say, oh, I had that bismuth a year ago. Don't worry, you can have it again because bismuth doesn't cause resistance. Similarly, amoxicillin, tetracycline, although you can't give that to children or pregnant women, so be a bit careful with tetracycline, and furazolidine. So these antibiotics almost never cause resistance to Helicobacter. So I mean, they are good antibiotics to keep using in combinations. So, but you only use these drugs once: metronidazole, clarithromycin, quinolines, levofloxacin, rifamycins. That's a TB drug, so a lot of people like to reserve rifaximin and rifabutin for tuberculosis. So that might not be a choice in most hospitals. They're quite expensive, I believe. So H. pylori almost always becomes resistant to these, and there's a high resistance rate in China. We know why that happens. H. pylori has a lot of mutations, so that can happen. So here's a new diagnostic tool as well as the breath test. We're going to use a non-invasive test, the string test. I'll show you a picture of it in a minute. So we've got non-invasive, UBT, fecal antigen, serology, the pros and cons of all those tests. The string test, you see, we have a string test which is 70% of people can culture H. pylori from just a swallowed piece of string. qPCR, antibiotic sensitivity, can be done on the DNA on the end of the string, and can be done actually from the culture very, very accurately. And possibly in the future, we may be able to get pathology inflammation looking at the genes expressed in the stomach on the string. And so that would mean histology culture and rapid urease test not needed. So how does string test work? Here's a piece of string with the Helicobacters on it. And they're very sticky to string, and they will jump off the wall of your stomach and easily stick onto the string. So we find lots of DNA then when we pull the string out. And so people have been using string tests for about 100 years. And we were using it in Australia, in the Australian outback, to look at H. pylori in Indigenous people 20 years ago. And we could do PCR and culture from those strings, etc. And there's a bit of data coming out now where we've done string tests in thousands of people, and we're trying to see the accuracy compared with the breath test, for example. And it seems to be very similar. And so in this particular study is a couple of string test publications. So we'll just look at this 2x2 table, see? So if the string test was positive, all the breath tests were positive. So that's good. We know it's positive predicted value is pretty high. So if the string test was negative, the breath test was positive in 52 out of 58. And also I'm trying to work out what the numbers are. But you see, if the breath test was negative, the string test was positive in 6, where the string test was negative. So does that mean the string test is less sensitive than the breath test? Could be. Or it might mean that there are false positives with the breath test. So when comparing the string test with the gold standard, you really have to do, you do your breath test, and you do your biopsy, and you do your urease test, and you do your culture. And then you've got the gold standard. And you say, okay, now we understand why we're producing the gold standard, and we'll get the answer. We'll probably find there's a few false positives with the breath test, and maybe a few false negatives with the string test, because let's talk about people. Practicalities are some people didn't fast very well, and they've got some food in the stomach, and so the tests will all be faulty. So just remember that. I think we're pretty much. So I just want to say that qPCR on a string test is very interesting. Every hospital in the world has a qPCR machine because we do COVID tests with it in 4 hours. So now you can do a sensitivity test on a string test in 4 hours, or typically the next day, and start treatment. And you can give a person precision medicine. Now, as a gastroenterologist, you know it's very difficult. You've got a patient with resistant bacteria, but you're going to give a very difficult, PCAB, a quadruple therapy maybe with a lot of side effects. But if you knew that that patient had an organism that was sensitive to metronidazole, you say, whoopee, good news. We're just going to give you a 7-day treatment with a metronidazole combination, and we should cure you 90% of the time. So that is great value for the string test to get that extra information. This is a qPCR machine, and you should all go and see how that works because they're very, very useful. So who would need to do it? A person who doesn't need another endoscopy. That's the main thing, or they have trouble accessing endoscopy, they're recently endoscoped, etc. And here's the strategy. So after you treat somebody, you do a breath test. If they're negative, that's good. If they're still positive, you do a string test as well. And then you do a qPCR, and you want to do personalized treatment. So if they've got a sensitive organism, that is great, easy therapy. If they've got a resistant organism, well, then you're going to go in with your quadruple therapies, high-dose PCABs, 14-day courses, etc. So not everybody needs to go up that pathway. Some people go down here. And so I think that's where this is going to come into its own in the coming years. So finally, I'll just acknowledge my colleagues. My important colleague is my wife, who helps me write all my papers. She's very, very literate. She's much better at writing and literature than me. And so her name is Adrienne, and she still publishes as a co-author sometimes. She did one recently on that eulogy for Dr. Warren. And there's Dr. Warren there receiving the Nobel Prize, and our associations and affiliations are down there. Sorry I took so long, but I think everybody was still watching the lecture, so I hope they didn't go to sleep. Thank you.
Professor, it was, we were humbled by your talk. It was elaborate, excellent, and extraordinary. Now, all questions will be like a rapid fire for you now. The panel's discussion starts now. The first question is by my senior colleague, who is the director of Institute of Liver and Biliary Sensor, Dr. Sareen. Dr. Sareen, please, you can unmute and ask this question. I've just projected the question which you have sent to us.
Thank you. It's a privilege for us that we could hear the real story from the absolute salutary work that you have given to all of us. Briefly, my question has been partly answered by you, but is still, as a translational scientist, I want to know the role of power of observation and conviction vis-a-vis serendipity in the discovery of H.Pylori as the cause of gastritis and peptic ulcer. Had it not been a weekend, had it not been you were so observant, would we still had the chance of having H.Pylori?
So Dr. Warren reminded me of a quotation from Louis Pasteur. And it's in microbiology, this kind of thing happened a lot. It says, chance favors the prepared mind. And it's amazing how often that happens. And I can tell you, Dr. Fleming, who discovered penicillin, he also discovered lysozyme in the eye. So he was a person who was doing studies of inhibition of bacteria on Petri dishes. And so when he had a mold contamination on his Petri dish with penicillin mold, he came back from holidays, he saw a big zone around the mold. He says, the mold must be inhibiting the bacteria. You see, a lot of people would not even notice that. So it's the case of serendipity, if you like, but he was on the lookout for those kinds of things. So Warren was interested in bacteria. And of course, we had all been taught that the stomach was sterile, but then you do have to believe your own eyes. And so the answer to all these things, if you think maybe something is associated, we'll get more cases, get more data. And, this is what we like to see. I had some very hard lessons when I was negotiating many years ago with the FDA. And they would say, it's not proven, Dr. Marshall, we want another trial. We want more data. get more data. Everybody's saying to me, get more data, have double-blind data, get independent data. So the answer to all these things, particularly in clinical medicine and medicine, is start collecting data. And so that is the thing. If you think, as a doctor, every time a patient walks in the door, you quickly develop hypotheses as to what's wrong with that patient. And you say, well, that's interesting. I saw a patient last week that was like that. They were eating something and they had this symptom. So then you make a note. And these days you can keep your notes in your phone. You can continually collect a little database. And after 6 months, you look, I didn't notice, plenty of people were eating that kind of cabbage, had no symptoms. So probably not that. So you can develop your own hypotheses and then do your own observations, little clinical trials, and that's how you develop a hypothesis. And maybe that's a lead. So then you would try and do a formal study. And so originally you might say we got only 20 patients. We call that a pilot study. But, that information gives you some a way of making a plan, if you like, to do a proper study. There's no point starting off a study on 1,000 patients and you haven't tested the protocol or you haven't tested the pathology stains. You need to get all that working first. So it does take a little bit of time, but have a good medical school and a good science teaching will have everybody understanding this. You have a hypothesis and you test it. The thing about a hypothesis, a good hypothesis is testable. If you cannot test it or develop a strategy to prove it wrong, then there's no way of testing it. So it's not a good hypothesis. And you will not get published in my journal. You may get published in a cosmetic journal, but not in a good journal like the Lancet or the New England Journal or whatever. I'm a little bit suspicious of the Times of India publication about that cancer data. It seemed to conflict with some other. And we'll have to discuss that again in the future. But anyway, great question. And I say, people who ask me, Dr. Marshall, how can I get a research success?
Dr. Marshall, you have actually answered in a very simple way to me, is that you have to have a prepared mind to accept observation. And I think that is a good message for me and maybe many of our colleagues. Incidentally, there is something green behind your back. I don't think it is H.Pylori. Is it H.Pylori?
This one, yes, it's H.Pylori. There it is. It's a H.Pylori model.
Yeah.
Thank you,
I'm
Professor
pleased you saw
Marshall,
that.
and thank you, Dr. Sarin is our senior medical scientist of India. Thank you, Dr. Sarin, coming over, and stay on with us for other questions. Professor Marshall, it looks like the weekend seems to be very crucial, both for penicillin and for H. pylori.
Oh, yeah. So that story that over the Easter weekend in Australia, we have a Good Friday holiday and Monday holiday. We're very lazy. So the microbiology lab never looked at the research samples over on Saturday, and it was day 5. And we now know it takes 5 days to grow H. pylori, not 2 days. And but however, we were trying how to grow H. pylori for 6 months. We were trying all kinds of media, microaerophilic, so that we had the prepared mind. And then finally, when we figured out it took longer, after that, it was like opening the instruction, the cookbook, if you like. And after that, everybody could grow it. So it happened with us as well.
Something like akin to that happened with Alexander Fleming also. So we'll move on to the second question, Professor. Dr. Reddy is our pride, like I was Dr. Sarin. His question, I think he would be asking you directly. He is the chairman and the senior most gastroenterologist of Asian Institute of Gastroenterology, Hyderabad. He had done a great endoscopy service to the humanity. Dr. Reddy, please.
Thank you, TSC. Thank you, Barry, for that fantastic journey and the lecture that you gave. Of course, we are looking forward to your physical visit to Hyderabad in November. My question is regarding the incidence of cancer stomach. I know that you already showed some variable data which of course, we still don't have the actual data, but in general, as gastroenterologists, we have seen cancer stomach does not parallel the incidence of H. pylori. Is it because we have a gastric mucosa which is different, or is it because of H. pylori? There's some work to suggest that we have a less virulent H. pylori in Indians. What would your opinion be?
So I think it's probably two parts to it. Well, you get gastric cancer when you're older, and so I know that the average lifespan in India now is 70. Is my recollection. So there's not as many old people. If you look at the data around the world that of the gastric cancers, for instance, in Japan, you know, everybody lives a bit longer in Japan, and so cancer rises exponentially after age 60. So people at the age of say 70 or 80 in Japan, they would be getting like 20 or 30 per 100,000 per annum. So you've got to do some correction of the statistics that you're looking at. That's one thing. Second thing is, you're right. The H. pylori in India, some of it doesn't have the CAG. So in Australia, I think 60% have the CAG toxin. And then when it does have the CAG toxin, there's different varieties of it. And the strong CAG-A it's called type D, that's really the optimized one for inflammation. And so that's 100% in China or 96%. In India, that's probably about 50% of strains, but I'm not sure. And actually, India and Europe, some of the strains don't have a very strong CAG-A, it's weaker. So that's possible. So the next thing is the food. So I'm not sure how it could work, but it's quite possible that there are things in the Indian diet which inhibit H. pylori a little bit. Now, I have seen data in gerbils and data in Korea on lactobacilli and different kinds of yogurt, where they try to find a yogurt that inhibits H. pylori. So it doesn't cure the H. pylori, but in one animal model, in gerbils, they found that the gerbils were getting less cancer if they were drinking lactobacilli compound. And in vivo, they could show lactobacilli was sort of killing a few helicobacter. So I'd like to see better studies on that kind of thing. And I know it seems to me that it's quite possible that some of the vegetables that Hindu people eat all the time in India would inhibit H. pylori, which is good news for me, because I went to a conference in Bangalore, it was all vegetarian, and I thought, oh no, I'm not getting any meat. But it was quite delicious, actually. So I'm not frightened of eating vegetarian food anymore since that visit. So you can educate me some more. But certainly, actually, one of the other things that's a bit difficult to test is measure the H. pylori. And so I'd like to see people do a few studies on how you could accurately quantitate the H. pylori in the stomach without actually doing biopsies. And so I would suggest that you could take a breath test with a meal, and you could roll the patient this way and that way, and then take breath samples every 5 minutes out for 30 minutes. And you could say, people have lots of H. pylori or not so many, because there's so much variability in the breath test amongst the positives. So C14 test, one day you're at 1,000, next day you're at 2,000, and we don't really know why it's different. It's because of the distribution and motility and things like that. So I would say that, so a bit of the host, a bit of the diet, and a bit of the organism. But I hope that I can see some good data coming from India. I think it's kind of an important question, and you could say, oh, this product or this diet really does protect you a bit from stomach cancer, inhibits the H. pylori. So since we can't possibly treat 500 million people with H. pylori just all at once, we could give everybody a bit of advice and just focus on the people with bad symptoms or bad history. So I think that's typically how it happens. And so good question.
Thank you, Barry. Thank you.
That we don't have that much of cancer stomach, despite having 50% of the Indians positive, as per the statistics for H. pylori, thing is protecting us from cancer. Can you move on to the third question from Dr. Kuru, who is again another senior medical scientist from India. He has done a lot of original work in the hepatitis cyst and hepatitis G. He's Dr. Kuru to you.
Good evening, and Professor Marshall, it's been an honor to listen to your journey. From Kashmir, we have a very high incidence of peptic ulcer and gastric and esophageal cancer. And over the years, we have proved it with excellent epidemiological studies and published it internationally. Now, over the years, the duodenal ulcer and the peptic ulcer epidemiology has changed. But what do you think would happen to a very high incidence zone of gastric cancer in Kashmir over the next decades with the changing epidemiology and current status of H.Pylori? This is in a very high incidence zone of gastric cancer, where the incidence is nearly more than 40 per 100,000 per year.
Wow, wow. So all I could suggest is that now Kashmir is probably a bit closer to the Chinese strains of Helicobacter, and so I would predict that you've got a higher percentage of the nasty D-type CAG-A in Kashmir. I think there probably is data there, but I didn't get time to look at it myself. I think probably that's what you would find. And then, what else could it be? I think just people catching that organism at a very early age. So I would think of maybe the dietary habits. In England, for example, people who had hot and cold water in their house were washing their hands, and the hygiene.
Very special dietary and personal habits, very distinct, and it's ethnically a distinct population.
So one of the countries where there's quite variation in the Helicobacter rates, for example, is in Malaysia, where the Muslims have much less, maybe only 30%. And in Indonesia, there are places with very low levels of Helicobacter. So it's not just socioeconomic, it's the quality of the water and habits. I know there's a lot more hand washing in Muslim families, I suppose, as part of it is the rites. And then in Indian families in Malaysia, about 40% were infected with H.Pylori. And in Chinese families, it was 60%. And it's not socioeconomic because the Chinese families tend to be a bit well off in Malaysia. So the interesting features there, I think I would look at the diet, and I would also try to figure out when people are catching it, and do they have the same strains as their parents, or did they have different strains? Because if they have different strains from the mother, then it means they're probably catching it from the drinking water or something in the environment. They're interesting questions, and I think there's a lot of interesting research for people to do. So Kashmir, I know that the British Army used to recruit, I guess, the Gurkhas from the northern India. And they used to come down to southern India and get epidemics of duodenal ulcer, was my recollection. And so there was a lot of interest in India on diet. What was it? Were they having more meat all of a sudden, and they had the diathesis and they didn't make enough acid, and then their acid secretion increased? So things like that could happen. But I'll just tell you that some of the worst cases of duodenal ulcer that I have seen have been in Indians. And actually today, I got a letter from, I don't know whose patient he is, but because of the publicity going out from this webinar and my conference in a month or so, there's people sending me emails asking me about their duodenal ulcer. And I can remember, there was a patient, he was almost a patient of mine. I was helping with eradication, but he was a student. He was perfectly well, he thought. And he's just a medical student or something. And all of a sudden, he went unconscious. They took him into the hospital. They didn't know what was wrong with him, but obviously his blood pressure was low, and he didn't even have melena. But on ultrasound, they figured out that he had a bowel full of blood, and they opened him up, and he had a bleeding duodenal ulcer, and he just almost bled out, and they, caught it, did diathermy or something like that. and treated his H. pylori and he got better. But, these people, there are people who just suddenly die from peptic ulcer. And I probably, up in Kashmir, people see that from time to time.
So the peptic ulcer, the incidence is 4% in lifetime, prevalence about 14% in Kashmir of peptic ulcer. Those are the data which are very epidemiological data here.
So it used to be much higher in the United States and in most countries. In Australia in the 70s. It's interesting to me that peptic ulcer in some countries kind of parallels cardiovascular myocardial infarctions. And so when I was doing my internship and training in internal medicine, there was a lot of relatively young men coming in and dying with heart attacks and also peptic ulcers. And there seemed to be a parallel, and it's faded out a lot now, because as soon as people get indigestion now, they can walk down they can get over-the-counter H2 blockers or a low dose of omeprazole. So they probably never get to those really bad stages that they used to.
Thank you,
but
Professor.
maybe it's changing.
We'll move on to the next question. Thank you, Professor Kuru, for the wonderful question. And we'll move on to the next question by Dr. Philip Augustine, another senior gastroenterologist from Kerala, the southern part of India. Dr. Philip, please.
Professor Marshall, there is some concern that gastroesophageal reflux may increase if H. pylori is eradicated. Since there was no scope to discuss that in your lecture, what is your opinion about it?
So in some individuals, I agree that it does unmask quiescent GERD. And so your question is actually already has the answer in it. So so years ago in Holland, they gave people proton pump inhibitors. So they had H. pylori people with they were just people with H. pylori, and maybe they had GERD. They put them on a proton pump and measured the H. pylori. In about half the patients, the H. pylori became difficult to detect. So if there's a high pH, there's so much ammonia around the H. pylori, it inhibits the growth of the organism, and your biopsy might be negative and your breath test might go negative. So that's why when you're doing breath tests, you always stop the PPIs for at least a week, probably 2 weeks if possible, before you do the breath test to test for H. pylori. Otherwise, the H. pylori can be suppressed by the inadequate acid secretion. So it's kind of a paradox. But on the other hand, some people actually have higher H. pylori levels. And so they might have like a duodenal ulcer distribution where the H. pylori is mostly in the antrum. And when you remove the acid secretion, the H. pylori goes up and colonizes the whole stomach up in the corpus. You see, so that people who have GERD, when you look at a population, maybe they haven't got GERD. Some of them will get better because when you treat the H. pylori, acid secretion drops, nocturnal acid secretion drops by about 50%. So these people have hypersecretion of acid during the night. And so those people, they might have a bit of GERD and they might get better because the acid secretion goes down. Other people have got low acidity and they might have a low acidity from the H. pylori and they have a hiatus hernia or a loose sphincter. Maybe their work is bending over a lot. And then suddenly they might have the hypochlorhydria a bit from the H. pylori. They might have bad corpus gastritis and not much acid secretion. And then you turn them into a normal person with normal acid, and then they've got a leaky sphincter. And so they have GERD. So it can go either way. And I would say if you've got upper GI symptoms with H. pylori, let's treat the H. pylori and get the mucosa back to normal and see what happens. And if it unmasks GERD, don't worry. We're going to put you on a PPI anyway at that point.
Thank you, Professor. We have another interesting question from the Professor of Gastroenterology from All India Institute of Medical Sciences, our premier medical institution in India. Dr. Pramod Gar. Dr. Pramod Gar, please.
Sorry, Dr. Chandrasekhar. Can this be the last question for Professor Marshall?
I have two more questions. Okay, no problem.
We'll
Dr. Pramod Gar?
do two more.
Okay, two more.
Two more, okay.
That's it.
Okay. They're good questions.
you.
They're good questions.
So thank you very much for this. Thank you very much for this brilliant talk. We are all humbled. So my only question, which is rather non-medical, is you have, of course, achieved huge intellectual satisfaction through your excellent science. How about attaining nirvana, spiritually speaking? Thank you.
Well, I would say the nirvana is making a discovery, and most people would say that when you make a discovery and you make an observation and you realize that you have got some new knowledge which nobody else has, and you can then dream and extrapolate so many different possibilities if you just by sitting doing brain experiments. And I suppose the classic of this is Albert Einstein, and he's walking his dog, he's thinking about stuff, and he does Brownian motion and black body radiation and relativity. He thinks it all up, and he never ever went into a lab. He never did any experiments. He just looked at other people's data and just thought about it. So, I mean, that is a beautiful thing when that happens, and that did actually happen with Warren and me. Once we started looking at our data, you can imagine our mind was just already in nirvana, and we're very lucky that we're kind of lucky that so many people had H.Pylori. We couldn't have possibly dreamed of it. Thank you.
Thank you.
We can take it that your work can lead to nirvana.
Yes.
Thank you.
Yes, I suppose so.
Thank you. Good.
Thank you, Professor Garg and Professor. The next question is, another premier institute, from India, Postgraduate Institute of Medical Education and Research, Chandigarh. Professor Datta, please.
Oh, good.
Thank you, Dr. Chandrasekhar. Thank you, Professor Marshall, for sharing with us the excellent journey which you had with H.Pylori. My two quick questions are role of the coccoid form in
Can I request you to ask only one question? First question, Professor. Okay.
The role of coccoid form of H.Pylori and the role of biofilms with Candida in causing refractory H.Pylori infection and gastric cancer.
Okay, the first one is very easy. When we eradicate H. pylori, it is 100% gone. You cannot find H. pylori anywhere in your body, and usually people don't catch it back. So the mystery is, I had H. pylori, I keep taking the treatment, and then I get it back again. That is not really true. That treatment did not work. And so usually treatment is 2 weeks, and then you wait a month, and then you'll repeat it. If you have dirty water, like in Peru, the water's contaminated, 15% of people will catch H. pylori again in 12 months. So everyone's say not worth it. But, if you have ulcers, it's worth it. But normally, in Malaysia, in most countries, the reinfection rate is 1.5–2% per annum. So usually it's failed treatment. And the biofilms in H. pylori, I'm not an expert on that.
Okay, right. And okay, now the last two questions. You want to say something about biofilms and H. pylori, Professor?
Oh, yeah, well, you do have biofilm on the back of your tongue, which has got a lot of candida, and people can have H. pylori in their mouth, but it's just a transient contamination from a reflux episode. We don't believe it lives in the mouth. We can't find live ones. Okay.
Okay, so for the senior medical, the scientists were asking, one student, young medical graduate from USA wants to pose this question. Maybe you achieved a lot as a medical student and taken to the level of Nobel Prize. So his question, Dr. Vivek, you want to answer this question? Ask this question?
Yes, thank you, Dr. TSC. Professor Marshall, thank you very much for such an inspiring talk. When you started your career, you probably didn't have an idea that you were going to get the Nobel Prize, but at some point during your journey, you probably had that inkling, given how much it revolutionized the management of peptic ulcer disease. So my question is, if someone were to have that, you want to achieve something like getting the highest honor of the Nobel Prize, what is your suggestion or advice regarding the pathway a clinician should take?
Okay, so I would say this is actually a formula for failure as well as success. But take the difficult tasks. Try to solve the difficult problems, because you will have very little competition. Most people take the easy path, so you will have little competition. If you succeed, you'll be like the conquering hero coming back from the battle. You see, you won. And so you don't, then the Nobel will happen. So that's it. So when you're thinking about your research and research goals, you've got to think over the horizon. So what's over there that I could discover? What would I like? What would I imagine we could do for this disease or this illness? And you've got to think of something where people are no longer there. People just extrapolate from their current knowledge. So you got to say, okay, what's that going to mean? Go 3 or 4 steps ahead and think about what it could mean. And then you can actually express that in your research grant. You say, well, this might seem a boring thing, a few bacteria, blah, blah. But if it turned out that it was very common, maybe it could be an important thing. And I know a lot of people are thinking that nowadays, but you jump ahead a few days, few generations, if you like. And so many things. And the other thing is people who think the illusion of knowledge. If you can find the bits of knowledge that are not based on science or fact and prove them wrong, that's a good start. And then you might open up some other thing. And as a young person, if you're connected to the lab or doing research, do it well and make sure that your technology and your techniques are beyond reproach. And then if you get funding, you can take your funding and go to anybody's lab in the world. And so I want to go to Stanford, to that chemistry Nobel Prize winner's lab. And you call him up on the phone. And the first question he asks is, have you got your own funding? And you say, yes. And he says, okay, you've got a job. See you next week. So that's how you do it. And so don't be shy about coming from, wherever. I mean, you've actually been in Augusta University, USA. So I think you learn this in America, don't you? And you don't necessarily, the other thing I would say to people is that all humans are pretty much the same. And so if it works in your little hometown in the middle of India or in Western Australia, then there are millions and millions of people who really like to have it.
Okay, Professor Marshall, we have only 1 more minute. You want to take up the rapid fire as rapid as possible. And list view is watching me. I know that's a little upset. But what is your favorite non-medical book? This is just to know about you. That will inspire thousands of doctors watching you. What is your favorite non-medical book?
I think Tom Clancy's book, Red Storm Rising. And that really preempted the end of the Cold War and the USSR and things like that, and had a lot of things in it that we didn't dream of, like stealth technology and HIMARS, all those technologies. So it's taken 30 years to see those everywhere. Okay. But I think he writes a good book, and it's good for a long plane flight from Perth to England or somewhere.
Probably when you want to come to Chennai, you can read that. We can read that from Chennai. Who is your hero in life apart from Professor Warren?
Well, I had a number of medical heroes, and as a young boy, I used to read, the biography of Thomas Edison. Somebody, and this concept of making inventions and helping civilization. But medical-wise was John Hunter, and there's a lovely book called The Brother Surgeons, and it tells you the story of his brother, William Hunter, who was a fancy, rich surgeon up in Edinburgh, and John Hunter was the youngest brother, and they said, "Oh, we're going to put you in the army." So he was just a tough, rough army surgeon, and he made so many great discoveries. And you can visit John Hunter's museum in London. If ever you go to London, you must go to his museum and see all the stuff that he collected. But Brother Surgeons, read that book. It's a great fun book.
I was fortunate to visit that museum. I was lucky. And what is your favorite quotation, which really inspires you every day, or what is your favorite?
My favorite what?
Quotation.
Quotation. Okay. So one that I like, and I don't know who said it first, it wasn't me, but science is not a democracy. So it doesn't matter how many people vote against your discovery. If you've got data, you're the one who's going to win out eventually. And I can't remember, maybe it was Archimedes, he said, you know, give me a lever long enough and I can move the world. And someone else said, if you've got the correct facts, you can just change the world.
I think I like
if they don't have it.
Science is not a democracy. I think that's a great quotation. If you are having conviction and you are confident, you will always win the race.
Yes, yes.
Who is your favorite sports personality?
Well, he's pretty old now, but he's my vintage. But Dennis Lilly, he was West Australia's famous fast bowler, and he used to get into fights with the Indian teams because if they were great batsmen. And I remember he tried to introduce the aluminium cricket bat. And I was very controversial. That's when I was doing my internship, and all the doctors at Royal Perth Hospital used to neglect the patients and watch the cricket on TV. The Dennis Lilly was a really famous fast bowler.
Oh, great. And who is your favorite film personality? Movie, film, cinema?
Movie personality, I suppose.
I don't think you have seen movies for ages. I think you've not seen any movie yet.
Yeah, well, it used to be John Wayne, I suppose, when I was a kid. But nowadays, I like a bit of action movie. Well, actually, I think Tom Cruise. I trust Tom Cruise. He's got some strange ideas about life, but actually, he knows how to pick good movies, and I can always trust him to have a good story.
I think he is favorite of many of our Indians also, because he doesn't have he does the stunt himself. He doesn't believe anybody doing for him.
Oh yes, he actually had a lot of a lot of scenes recently in the Himalayas, I believe. That's probably up in Kashmir or somewhere.
Okay, the last one is, what is the mantra for work-life balance?
Well, you see, you want to plan your life and your research and your career and try to be sustainable from the very beginning. You know, when you have an exam or a big deadline or something coming up, you have to work, 18 hours a day and 2 weeks solid. So I have a bit of that coming up in the next month or so. But most of the time, I would say that you need to pay attention to your family as well, and realize that they are the people who are going to support you when your experiment fails. Your colleagues are just going to say, Barry, we're not paying you anymore. You haven't got a research grant. But your family will say, don't worry, you're really a good person. So you need that strong family. And I remember, I'll give you this quote from my Nobel Prize speech. Yeah. And so I said, work hard, keep balance in your life, and just in case, always be kind to Swedish people.
Very good.
So I'll leave you on that thought.
I think you have fulfilled all the questions. If I have missed out something, I apologize for that. And we were humbled by your lecture. It was elaborate, excellent, and extraordinary. It has inspired thousands of delegates from all over the world, not only physicians, pharmacologists, and the pathologists and microbiologists. And your rapid fire was much more rapid than what I thought. And it is fantastic. And the whole panel, I must thank that they are all from various parts of India. They are also pioneers in their own field. And they are the HODs and the department heads in various leading medical colleges in India. They could spare time. And I must thank you very much for this wonderful evening. I was told that you don't accept any lecture beyond 7 p.m. And we were very fortunate that you accepted. And it's a great honor. And a great honor accepting our award, which will be couriered to you. And we need a photograph of yours with the award and the medal. Thoroughly enjoyed. This is our first experience with the Nobel laureate. And I grew up with the thought that the inspiration to become a Nobel laureate. At least I could meet you, interact with you, and I'm getting more inspired. I will pass on this inspiration to the next generation. Thank you very much, Professor. Thank you.
Okay, thank you for all your preparation. It was a wonderful session, I think. Thank you. Good night.
Thank you.
Thank you.